Okay. Hello, everyone. We're going to continue with our second keynote speaker for today. So I am very pleased to introduce my friend, Dr. Rose Faghih, who is an associate professor at New York University. So Rose and I are both technical representatives for the IEEE Engineering in Medicine and Biology Society. So that was how I met her. And she is just a phenomenal lady. She's won many awards, and I thought it was interesting when I was reading her biography. It said that in 2020, she was featured by the IEEE Women in Engineering magazine as a woman to watch. And indeed, Rose is a woman to watch. She won the Early Career Achievement Award for our society, IEEE Engineering in Medicine and Biology Society, and as well as being the 2023 National Science Foundation Career Award recipient. And I mean, I could just spend like 10 minutes reading through her achievements, but how about I just let you hear from her as what she's going to talk to us today about computational medicine in mental and physical health. And I just want to encourage you again, please, after her speech today, take some time to get to talk to her one-on-one. Rose, join me in welcoming Dr. Faghih.
Thanks a lot for the kind introduction. It's an honor to be here. So I'm going to talk to you about some of areas of my lab's research focusing on developing algorithms for mental and physical health applications. To just give you some background, I was trained in two different labs. So one of the labs at MIT in electrical engineering department focuses on information and decision systems and controls. And then the other lab focuses on neuroscience, medical applications, and statistical signal processing. Then with that background, when I set up my own lab, I decided to focus on wearable technologies as well. So basically, the goal of my research is to come up with cutting-edge solutions for healthcare applications. So in my lab, we work with dynamic data, which is pulsatile. So you can think about the different hormones as basically biochemical signals that are highly fluctuating, and there is information in the pulsatile signaling. Also, we work with bioelectrical signals, for example, skin conductance response, which has a high level of fluctuations. And there is basically information in this pulsatile signaling. So what we do is we develop algorithms to take these pulsatile physiological signals and infer the underlying neural activity, which is basically transmitting the information. Once we have inferred that information, then the next step is we build these decoders to estimate hidden states. They can be related to our physical health. So you can think about, for example, hormone, cortisol, and then you can look at fatigue, or you can think about cytokine data, and you can think about decoding inflammation, or we can think about skin conductance data and decoding one's cognitive arousal or behavioral data to decode one's cognitive performance. So basically, with these methods, we build virtual sensors with our algorithms dynamically. And then the end goal is, okay, if I have decoded these hidden states, then I want to be able to do control and come up with different, basically, interventions to regulate these hidden states. So one application area that we focus on in the mental health domain, I call it a mind watch. So this is designing algorithm for future, basically, smartwatches that can infer brain activity and regulate brain states. So one of these, basically, brain states that we focus on is cognitive arousal. So we have been building decoders using skin conductance response, as well as, for example, heart rate or other physiological signals to decode one's cognitive arousal state. Then, once we have decoded the cognitive arousal state, we have been also doing experiments in our lab. So our lab is a computational and also experimental human subject experiment lab. So we have been using simple interventions like music or drinking coffee to improve basically performance and regulate one's arousal level. We also focus on other basically states related to mental health, for example, emotional valence or interoceptive awareness state. Then On what I call a health watch vision, we focus on estimating physical health states. So if you think about the glucose sensor, you have to collect a small amount of blood to basically measure glucose sensor, basically in a variable study, but you cannot measure something like cortisol hormone in basically this variable setting. Now there's a new technology from Bristol called Uritem. So it is basically a wearable device that you can wear over 24 hours and you can collect cortisol data dynamically. It is big and bulky. It is not practical for everyday use, but they are working on basically making a much smaller sensor. So we take either clinical data, which is dynamic, or Uritem data, and then we build algorithms to recover the underlying pulsatile secretory information, and then we build basically different decoders for decoding fatigue, inflammation, and metabolism. So for the first application area, which is mind watch, we do experiments in the lab to close the loop. But for the health watch domain, basically we do in silico studies. So we develop also a lot of control algorithms to develop these closed loop systems. But then the vision is once basically these sensors become very small, we can have a wearable device that can hopefully dynamically collect all of these data, and then basically in a real world setting, we can use our algorithms and can close the loop. So that basically there's a difference between the mind watch and the health watch directions in that sense. So for the mind watch application domain, one application is optimizing productivity in smart workplaces. The other application is maximizing cognitive engagement and learning in online and in-class environments. Aging in place and pain regulation are other application areas for this research. In the health watch domain, one application area is tracking fatigue. So if you think about chronic fatigue syndrome, fibromyalgia, this has been a challenge for a lot of women, but it didn't impact men. So now, if you think about long COVID, one of the symptoms that, regardless of gender, basically these patients struggle with is this high level of fatigue. So one application domain that we focus on is basically building decoders for decoding fatigue. Now let's think about when the pandemic happened. So when the pandemic happened, they were trying to figure out who was at risk to get hospitalized. And for a lot of places, they decided, okay, like elderly should be hospitalized, and then younger people were dying. The reason is you cannot measure inflammation. Basically, it's a hidden state. So one of the things that we do, we are working with cytokine data to decode inflammation. So basically, you are building a virtual sensor for decoding inflammation. And then you can have other applications, for example, in critical care unit. Another application area for this health direction is tracking hunger and energy, which is related to obesity. And basically, the end goal is to be able to decode and track these hidden health states to enable at-home remote health monitoring and also enable basically healthcare for everyone. So really, the focus of our lab's research is to come up with algorithms for next generation health-aware and brain-aware wearable technologies to basically dynamically infer and track hidden health states to then come up with interventions accordingly. So basically, if you think about, if you want to measure brain activity and look at one's cognitive arousal, you can very easily do that using EEG. However, let's say someone has a mental health problem. It is not practical for them to basically wear an EEG headset and walk around. So now, if you think about basically a smartwatch, it is very normal to wear. So then the idea is that, can I get the same type of information that I can get directly from the brain using a smartwatch? So how do we do this? One signal that we focus on is called skin conductance response. Has anyone heard of skin conductance response? So what happens is, when someone gets aroused, because of the changes in the arousal, there are these micro fluctuations happening in the sweat activity. These are very, very small, so we don't feel them. But if you think about our skin as an electrical circuit, so the resistance of our skin is changing. Conductance is 1 over resistance. So over here on top, you see the skin conductance response, which is highly pulsatile. So these arousal events are basically encoded in that pulsatile signaling in that signal. So what we do is we come up with physiological models to basically model skin conductance response and infer the underlying arousal events. So basically, we infer the underlying arousal event coming from the brain using the data we collect from the skin. So it can be a wristwatch, it can be from the foot, different basically locations. And then once we have done that, we build our decoders to decode the hidden cognitive arousal state. And then the last basically step is to come up with controllers. So basically, we enable a smart watch become a brain-computer interface that can give us basically the information about the brain, and then we can regulate that brain state. So to just show you that we have the ability to get some basic information about arousal level, so we did this experiment where we have a low arousal task followed by a high arousal task. We collected skin conductance response as well as basically FMIR's data from the prefrontal cortex. So basically using FMIR's data from the prefrontal cortex, you see we have like a low level of activity during the low arousal task and an increase in the activity during the high arousal task. And then when we build our decoder and then we define also this arousal index, we detect the low level of arousal during the low arousal task and a high level of arousal using the high, basically arousal task. And here, as you can see, so we have done this study independently of each other. Then the next part is, can we actually regulate brain states like something like cognitive arousal using something as simple as music? So we did another experiment that I show you here, where first the subject is at rest. Then this subject has fear of heights. So we basically show them videos that result in a fear of height response, a fear response in them. So you can see their skin conductance response is going up as a result of basically this stressful video. And then we start playing relaxing music for them in the second half of exposure. As you can see, basically, based on a decoded state, we are lowering the level of arousal. And then when we stop the exposure to the stressful video, we continue relaxing music exposure, we see that we can lower the level of arousal even further. So something as simple as music basically can regulate our cognitive arousal state. So in terms of the algorithms that we developed for this, so if you think about these arousal events coming from the brain, and then we have our observations, our skin conductance response observations from the skin, you can think about a convolution operation is happening, and then you can have multi-channel data, so you can collect the data from the hand, from the foot, or from other locations. So basically, we have been developing these optimization-based and Bayesian-based, basically, deconvolution algorithms to recover the underlying arousal events. So here I'm showing you an example data set where we have the ground truth information. So the subject was basically exposed to auditory stimulation, which is disturbing to them. So you see that in gray. And as a result of that, we see skin conductance response. So we have, with our algorithm, we convolve the data, and as you can see, with a small time delay, we are detecting these arousal events. And basically, the idea here is that we are just modeling this based on the physiology, and we are utilizing compressed sensing methods because of the sparsity of our data. So without putting any assumption on the amplitude or on the time interval or basically any other constraints, we can just use the sparsity of this data to do the modeling and recover the underlying arousal events. And this is an example optimization formulation that we use for basically recovering underlying arousal events. So if you think about this type of framework, as we add more data, we can do a better job in recovering the underlying arousal events. So here we have hand data. Hand data is high quality because we have a lot of sweat glands in our hands. Food data is not high quality because we don't have as many sweat glands in our food. So now, if we analyze the data, hand data separately versus food data separately, so this is a simulated data set. The previous one that I showed you, the ground truth information, was basically an experimental data set. So here, for the simulated data set, in terms of the error, you can see that we can do a much better job with the hand data compared to the food data. But when we add the food data to the hand data and do concurrent deconvolution, we can basically reduce the level of error and improve our inference. And in comparison with what is done basically in the literature, so one of the issues that the existing methods have, they overfit, and they are not properly including the sparsity, and also they only work on single channel data, while having multi-channels is going to help us with the signal processing. So now, once we have recovered these underlying arousal events, the next step is for us to build our decoder. So for building our decoder, we use different types of observations. So one type of observation is, did I have a skin conductance response, or did I have an arousal event or not? So if basically I'm looking at if I had an arousal event or not, I'm creating this binary sequence. So it's a point process, basically, data set. Then I can think about it differently. I can say, okay, if there was an arousal event, I care about the amplitude, because if the amplitude is higher, it means that the subject was basically aroused more. So then, basically, you're going to have marks associated with the point process. So we formulate a marked point process observation. Then, besides that, we can have continuous data. So basically, we have a combination of point process, mark point process, and continuous observations that we utilize for building our decoder. And then basically we use this Bayesian filtering approach. So we have this expectation maximization algorithms, different versions of them that you see basically the QR code if you want to get the basically GitHub code or the papers here. And here you see example, basically, decoders that we have developed using skin conductance response and skin conductance and heart rate data. So I'm going to just show you two example decoders here. So one example decoder here, we are going to take just the marked point process information that we are recovering by basically first estimating these arousal events, and then we have the intensity of the arousal event, which is the amplitude. So we assume we have a random walk model for our cognitive arousal state. And then, if you think about our data, when we want to formulate this point process observation, we are going to select the bin size such that we have the largest bin with no more than one response in every bin, so that it is going to be basically a point process binary data. You can think of it as a coin flip with a probability qk, I'm going to get a response. And now, I model this as this basically zk, which is my arousal level, goes up, then the probability of having a response is going to go up. So it's dynamically basically changing. Then in terms of the amplitude also, we model it as if the level of arousal goes up, then basically this amplitude is going to go up. And based on this model, then we develop our expectation maximization algorithm. So here I'm going to show you one example experimental data that we analyzed. So if I give you instructions and I ask you to perform a task, do you expect to have a high level of arousal or a low level of arousal? High level of arousal, right? So here we have the skin conductance response. The first task is instructions. As you can see, we have a lot of skin conductance response, and then the decoded arousal level, E, and then F, which is the high arousal index, are both high, right? Now, the next task was the counting task. if I ask you to do subtraction, do you need to be cognitively engaged, or are you going to have a high level of arousal or a low level of arousal? So if you're doing basically a cognitive task for counting, you should be cognitively engaged. So in that sense, like, it should be a high level of arousal. But it depends. I mean, for some people, it might be easy. For some people, it might be hard, depending on like what it is. But basically, it's not like a relaxation. So we expect to have a high level of arousal, and then that is basically what we get based on the skin conductance response, based on the decoded level and the high arousal index. So has anyone heard of the Stroop test? So the Stroop test is a color word task. If I take the word hello, I paint it in red for you, I ask you what is the color, it's easy, you tell me red. But now, if I take the word green, it is associated with the color, I paint it in red, I ask you what is the color, it requires more thinking, because you see on the screen the name of another color. So the Stroop test is basically, you see like sequences like the green, red, blue, but you need to basically tell us the color of the paint, not the word that you see on the screen. So it requires also cognitive engagement. And as you can see, basically, in the Stroop test, also we have decoded a high level of arousal and a high arousal index. So if I ask you to relax, are you going to have a high level of arousal or low level of arousal? No. And then that is basically what we observe based on our data and our decoder. And then the last basically task for this experiment was watching a horror movie. So the subjects were not going to be quizzed afterwards. So if they're engaged, they might have a big response. If they're not engaged, they might not have a response. But like there is no way to know. So here we are basically decoding a low level of arousal for this particular subject. So now I'm going to show you another decoder. For this decoder, we are using skin conductance response. We are also adding basically heartbeat information. And again, our goal is to build a decoder so that eventually we can close the loop. So here, I'm going to assume a simple random walk model like before, and then for modeling skin conductance response, I am basically, taking my binary observation here, so the point process, unlike the previous basically model that I was taking a mark point process, I had a mark associated with every point process. Then I'm also obtaining two observations from skin conductance. One is a phasic component, which is basically functioning based on the height fluctuations in skin conductance response. Also, skin conductance has a very slow observation, that is called a tonic component, so we are modeling that. And then we are also modeling our heartbeat information here. To model the heartbeat information, we are using this history-dependent inverse Gaussian model. And basically here, when we are modeling the mean, besides looking at the time between one heartbeat to the next heartbeat, we are also including the level of arousal in our model. So basically, as our arousal level goes up, then our mean is going to also increase. And here we have formed a multi-rate observations, because for every 50 heart rate bin, which is a one binary point process, we have basically one skin conductance point process observation, and we develop our decoder accordingly. So here we have tested this on fear conditioning, fear extinction experiments, which I don't know if anyone is familiar with that. This is like a task that is used in psychology and psychiatry. So we have two colors, which are two cues. One color is blue, it's a safe color. So it's a CS minus. So if you see basically the color blue, nothing is going to happen to you. Then we have the other color, red, which is a cue for a potential electric shock. So 15% of time, you get electric shock. So then you learn to fear, basically, that color. And here we have used our decoder, which is based on skin conductance and also heartbeat information. And as you can see, when the subject gets an electric shock, we are decoding a much higher arousal state in red compared to when there is no electric shock. Um, so now we go back to what I showed you in the beginning. So this experimental figure on top is basically the experiment that I mentioned. The subject had fear of heights. But then the way we did this study, we first start with modeling skin conductance, inferring the arousal levels, building our decoder. Then we came up with a supervised control approach. So we are using optimal control methods like LQR or model predictive control, and we build this fuzzy level on top. And then we do this in silico study first. So you see our controller in the in silico study, and then we did the follow-up study experimentally. So for the MindWatch application, when we develop our algorithms, we can also validate with experimental, investigations in healthy human subjects, also in patient population. So studying arousal is important for investigating performance. Let's think if you have a low level of arousal, you're not going to be engaged. You're not going to have the best performance. So now, if you also have a very high level of arousal, you're going to be stressed. You're not going to have the best performance. There is this mid-range, basically, for arousal that is going to give you optimal performance. So here, we investigated two types of music. One is relaxing, and one has, it's like calming music. The other one has like basically a high level of arousing music. And we want to see if we can modulate arousal to improve performance. And here we did this through this working memory task called the N-back task. Has anyone heard of the N-back task before? So basically in the N-back task, if you think about the one-back task, you're comparing what you are seeing now with one step back. If you are looking at like a three-back task, you are looking at what you see now with the 3 steps back. If you see the same letter, you say yes. Otherwise, you say no. So with that, I can measure the correctness of your response. I can also measure your reaction time. And both of these have information about your performance. So here, then we use our framework for decoding arousal. We also decode performance. And in terms of the type of music, majority of subjects perform better during the exciting music compared to the calming music, and they also perform better during the one-back task, which is an easy task, compared to the three-back task, which is a harder task. And then, so here you can see that we have quantified the hidden arousal state, and we have also quantified the hidden cognitive performance state. Again, from psychology, we know we have this inverted relationship between performance and arousal. And here, basically based on our decoders, we have quantified this inverted U law, and then we can use that to design our controllers. So here I'm showing you another experiment that we did this time. Instead of music, we used coffee. So you have skin conductance response there. You see basically the skin conductance response is decreasing. So we are detecting basically boredom, and then the subject takes coffee. If you take coffee in basically 8 to 15 minutes, you're going to have an arousal response. So then, if we wait basically that long, which is like subject specific, you see basically an increase in the skin conductance response. And then as a result of that, when we quantify the performance state, we can see we can improve performance. Of course, everything is subject specific here. We asked the subjects to bring their own favorite coffee to the experiment. So if you're interested in our experimental data, so it is publicly available. So we have research-grade data with music. We have also wearable data with coffee, music, and perfume. We didn't see much effect with perfume, but coffee and music work. Actually, AI-generated music worked better than coffee, and subjects selected exciting music, and then relaxing music was not as good as exciting music. So it's basically, there are like some differences, but they all kind of work. And then we also have another data set, if anyone is interested in downloading it, which is this variable exam stress data set. So we wanted to predict the students' grades using their skin conductance response. We did the experiment using the variable called empatica, which measures your skin conductance response. Unfortunately, we couldn't do any signal processing. We could just, because it was super noisy, because what happens is, if basically you think about your empathical watch, there are two electrodes that should be touching your skin. And if, like you are taking an actual exam, the students were taking like an actual exam in their circle class, you might not have that much contact depending on how you were wearing the watch. So the data was super noisy. You couldn't do signal processing, but with machine learning, would classify just high versus low rate using skin conductance response. And then this data set has like heart rate, movement information, and so if you want to download and like use that data set. So we have been developing a class of Bayesian filters. All of our code is available publicly, and then we have this tutorial book. So the first author is my former PhD student, and I'm the senior author. So basically, this is a tutorial book. So if someone is an undergrad, they want to learn how to develop these filters, or a first-year graduate student, they can. If someone, let's say, is in psychology, they can just get our code as long as they know how to use it. So this is basically a tutorial-style book that comes with code. This is publicly available, open access. So I have this flyer, so if anyone wants to take one, which has basically the information about like the QR code for downloading our book, downloading our code, and also downloading our data. So they're going to pass it around, and please feel free to take one if you are interested. So these filters can be used for other signals. Another basically signal that we have recently started working on is eye tracking. So the way we do this, we go back to the physiology, we come up with features that which are basically physiological observations that we take, and then with that we build our decoders. So skin conductance response has been used in psychology, it is like very well studied, and that's what we have been using in our lab. Then we came across this data set that included both eye tracking and skin conductance data, and they were doing the field conditioning for extinction experiment where you give basically electric shock, but the electric shock was very, very mild. It was not bothering the subject. So interestingly, we don't see that much basically skin conductance response. So if you're trying to here distinguish between red is when they were like given the shock, you see a very similar decoded state. Versus with eye tracking, it's more sensitive. Although like basically the electric shock was very, very mild, it can actually, when we decode the imperceptible awareness state, we can actually distinguish between these different trials that we have. Also, we have been building other decoders, for example, for investigating emotional valence. So if you want to understand someone's mental health, you have the cognitive arousal, which is basically the intensity of the emotion. Then you have emotional valence, which is positive versus negativeness of the emotion. So here we have been using a respiration belt data to decode one's valence level, and here we had subject-reported ratings as well. So if you think about subject-reported ratings, sometimes there can be a bias, because also like if you have subject-reported ratings and you have expert ratings, they do not match. So now your algorithm, for parts of it, they are matching, for parts of it, they are not matching, and it can just be because of maybe like the bias in the reporting, or if they are manually controlling the breathing, because breathing is very interesting that you can also control yourself. Like skin conductance, you cannot control yourself. But breathing, you can control yourself. So they do not perfectly match in like certain places, but that's basically something that you also have like subject ratings as well. Then in terms of emotional valence, we have done this in silico study based on experimental data that is using facial muscle activity, EMG, and with that we are decoding the valence level, and then we have been building control algorithms which are fuzzy based, so basically you are including decision rules based on the expert knowledge to then come up with the control algorithm to close the loop. Then under this mental health domain, We have also been using EEG data to decode how severe the seizure state is by basically decoding and tracking the seizure state. We have done like single channel investigation as well as multi-channel investigation. And the single channel investigation would be pretty useful if you are thinking of like real world basically studies and basically if you want to integrate it in the life of the patients. Okay, so now we're going to switch gears from the electrical signals to biochemical signals. So the vision is, if you think about right now the variables we have, we can easily, in a very cheap way, collect bioelectrical signals. Biochemical signals, on the other hand, glucose you can collect, but then all the other ones, it is very expensive to collect. So like if you want to collect data for 24 hours from one subject using Unicam, it's going to cost you a few $1,000s. And it's not real time. So, and then you get data like every 20 minutes. But eventually, hopefully, because now there is a lot of work, at least in the US, on sensing. So hopefully you're going to, maybe in the next decade, see some of these biochemical sensors, and then you can basically do both biochemical sensing and bioelectrical sensing to get the information about mental and physical health states. And the goal of basically designing these decoders, for example, a decoder for estimating inflammation or like fatigue, is to identify those who are at risk and then enable at-home remote healthcare delivery. So here you see the Unicam device that I mentioned, and we have been doing studies with this in the US. We brought it from Bristol. And this is basically measuring the data from the fluid under the skin. So basically in our modeling, because originally like we were working with clinical data, which was, the modeling was based on blood data. So now basically for this particular device, then like we have to change our model to include the sensor and then how the sensing is done. But cortisol is a signal that we have been studying a lot in our lab. So we have been working with different clinical data. This is like dynamic data collected over 24 hours. The clinical data is again blood-based. We also have basically real-world data using Unicam. So the subject wears it, they go home, they can sleep wearing it, then they come back, and then our nurse collaborator just removes it. Cortisol is a stress hormone. And basically, it is released as a part of this control system. But if you think about control theory, for example, if you think about the air conditioning system, you have continuous control. This is very unique because you have impulse-like control, you have pulsatile control. So there is this pulsatile signaling information there. And that is coming from the hypothalamus, which is right behind your eyes. So that results in release of the hormone CRH, which then results in release of the hormone ACTH from the anti-pituitary. So ACTH is right next to anti-pituitary is right next to the hypothalamus. So basically, right after CRH is released, ACTH is released. Then ACTH travels in the blood, goes to adrenal glands, results in release of cortisol, and we have a negative feedback effect. Basically, so if you have a high level of cortisol, then the cortisol travels in the blood, goes back to the brain and prevents further release of CRH from the hypothalamus or ACTH from the pituitary. So we have been doing a lot of like system theory modeling investigations of cortisol. And here I'm showing you like an example result that we have when investigating patients with fibromyalgia and chronic fatigue. So the cortisol clearance by the liver in patients is lower than the controls, and the number of hormonal secretion events during sleep in patients is lower than in healthy controls, and there is a lag also in the circadian rhythm of patients compared to healthy controls. So with this framework, basically, now we can study the healthy population and different disease populations. Then similarly, we have been developing these Bayesian filters for here decoding this fatigue state, which is related to your energy, basically this energy fatigue state. So here you're using a similar framework in terms of having the random walk model, but then besides that, you're also including the circadian rhythm because a lot of these models are physiological. And I should point out here for like the cortisol data, we have very small data because it's very expensive to collect. So basically you can collect maybe like 10 subjects and that costs basically a lot of money because you're doing this dynamically and there's information in that dynamic signal. So here the framework is very similar. Again, we are generating the point process data and you're modeling that. And then besides that, you're taking continuous observations. Here we are looking at the upper and lower envelopes on cortisol, which are basically giving us information about the circadian rhythm. So for this case, we are looking at the healthy population where basically we have a nice circadian profile when we take cortisol, and then we build our decoder, the decoded energy state as a nice circadian profile, so you have a higher level of energy during the day, lower level of energy at night when you're sleeping. But then if you think about a Cushing's disease patient, basically in Cushing's disease, patients have a high level of cortisol. So during the day, they are very fatigued. At night, they wake up because of their cortisol level. They don't have a circadian rhythm. And then when we decode the energy state, you see a high level of energy basically at night and a low level of energy during the day, which is matching what we know from the physiology in terms of the fatigue they have during the day and then sleeping difficulties at night. Again, we have been developing different types of filters. So another filter that we developed for this is a mark point process filter plus a continuous observation using the serum cortisol level. And again, we built our decoder, and we have decoded basically an energy state for the healthy subject, which has this circadian profile, versus the Cushing's patient does not have a circadian profile. And basically, again, we are observing a high level of energy at night, and that's why they wake up at night, low level of energy during the day. And then the end point with this is to just come up with the interventions also to help them with basically their quality of life. So we have the open loop case in the healthy subject that we have a circadian profile, there is no control. Then we have the closed loop case where you're taking two medications, you're modeling them. One medication is going to elevate the energy level, which is used during the day, and then the other one is going to inhibit release of cortisol at night so they can sleep. And basically with that, we have not been able to perfectly restore the circadian rhythm, but we have been able to obtain circadian rhythm in the Cushing model that we have. Then, we have been investigating other hormones. For example, we have leptin hormone, and then we have been building a decoder to investigate the satiety state. For this case, for example, we observed the satiety state during sleep is higher than during the wake period, for the satiety state increases after lunch and peaks in the middle of the sleep and is lowest in the morning. Also, the other thing is with a lot of these hormonal studies, we look at the causal interactions between these hormones, and it's interesting that for different subjects, it's very subject specific and can have different types of causal interaction. Then another signal we focus on is cytokines. So cytokines are very important in investigating inflammation. Here, similarly, we have been developing deconvolution algorithms to study cytokines, and we also have concurrent, cortisol data to study inflammation and to build decoders to decode inflammation state. So something that is very interesting is when we study the causal interactions of these cytokines, and cortisol. So patients with longer hospitalization had no causal relationship between cytokines and cortisol, while patients with less hospitalization time did. So there is information in that causal interaction that can help us, for example, how we think about the hospitalization time. And we have this other project, which is bringing together the electrical sensing with skin conductors and EEG with the urethane data for sensing cortisol to basically have bioelectrical and biochemical sensing together. So in summary, basically, we build algorithms to infer the underlying constant stimuli, build decoders, basically build these virtual sensors for sensing mental and physical health states, and the endpoint is basically also we wanted to control and come up with interventions to regulate these states. So for this to be implemented in real-world settings, we really need to think about the multidimensional, basically, aspects of one's mental and physical health. And also, we need to think about how, for example, environmental changes impact our brain model, our cognitive function, and then how activity can also impact basically these physiological signals. And with that, basically come up with these wearable devices for decoding and regulating health and brain function. With that, I thank you for your attention.